Hello, happy Monday. This is the 27th of January. It's try 2, week 10. Woo! Cells not actively engaged in the cell cycle are in some phase, and cells replicate their DNA in some other phase. If you have been missing class for the last little bit and you need to think about that, I think 4.6 is the part of AP Classroom Daily videos that are about cell cycle, and you can take a look at that. But also, I'm going to give you this little gift to help you know what that is. So it's week 10, and today we're thinking about the consequences of cancer's uncontrolled cell division. And remember, we are thinking about West Nile virus, but our case study right now is thinking about how we develop new cancer drugs and therapies and how we can look at data and figure out what broke in cells, because different medicines work in different people with different stuff that's broken in their cells. So today we're going to do investigations part 1 and part 3. If you have not been here, you're going to get some karyotypes. If you click through here, you're going to want to use the 2025 update, which is going to give you a typical karyotype for somebody assigned male at birth, a HeLa karyotype, the karyotype of a patient who has something called Philadelphia chromosome. And then I've given you that same picture and a picture of the cell cycle with all the different pieces. This one's going this way, but it's useful to think about many different representations. So both of these are going to go into your lab notebook. All right, our Unit 4 guiding questions, we're really thinking about the cell cycle here and how it aids in the conservation of genetic information. And we're also thinking about how we can use our understanding of cancer to develop equitable medical practices. So we spent a little bit of time together doing some claymation of this process, thinking about how diploid cells have 2 copies of each chromosome. This is a cell with 1 chromosome. Why are they colored pink and blue? Because one came from the sperm, one came from the egg. Families come in all shapes and sizes. And so reproductive technology even means that there are lots of different ways to think about that. So they duplicate during S phase and DNA replication. And then we marched through this process. On slide 126 or so is a movie to help you think about cell division. You can see some of it live. So we spent some time, I don't know if this is in this list, but we did it. We took a look at model 2 on cell cycle regulation. If you have not yet seen that, that's available in class, or you can read about cell cycle regulation in your review book, or you can watch 4.6 and 4.7. I'm trying to remember, but you'll go through, it'll say cell cycle and cell cycle regulation on AP Classroom, and you can watch those videos anytime. So I'm going to ask you to set up your lab notebook for investigation 7. If you're with the slides with me, it's going to be cell division, mitosis, and meiosis. And now we're going to do part 3. You're going to set up something called loss of cell cycle control in cancer in your lab notebook, and then you're going to put those paste-ins in. So we learned a little bit about this, and I wanted to show you karyotype. Karyo is from the Greek meaning nucleus. And these are pictures of mitotic cells where they've been stained with a material called Giemsa staining. Your chromosomes are not stripy in metaphase inside you. This is just a way that scientists have learned we can identify patterns and match up the chromosomes in somebody. So we're looking at karyotypes. You're going to review the cell cycle and checkpoints, and then we're going to make 2 claims and back them up with evidence from reasoning. We paused here to talk about the cell cycle one more time. If you have not yet, like I said, looked at model 2 on the cell cycle regulation POGL, I'd like to make sure that you do at least the first couple questions there or watch the video. You need to learn about cyclins and cyclin-dependent kinases. So what you're going to do is look at the karyotype. I'm going to ask you to link back in, go to those slides and ask this question: what checkpoint was broken to result in the creation of cancers with Philadelphia chromosome? So we take a look at that karyotype. It's up here. We gave you an arrow because you're not a medical professional. Hopefully it's pretty easy to see that when the cell was replicating its DNA, there was a change. The chromosome areas were really close to each other. And so as it was copying, that polymerase just kept making an extra copy, taking some of that DNA off 22 and ending up with a chromosome that has an extra little spot on chromosome 9. So that is enough to change cell cycle regulation. When a gene moves, how it gets turned on and off can change. And that is changes the cell cycle. So after you do that, I want to tell you about how a car starts speeding uncontrollably, right? You can brake the gas pedal, not BRAKA, but you can make it so it does not work. And so the gas pedal is on all the time. or you can cut the brakes so the brakes don't work. And we're going to apply this to the story of HeLa cells tomorrow. So the cool down today is to figure out the purpose of checkpoints in the cell cycle and be able to describe that in one good sentence. All right, that's it for me. I hope you're doing well out there. See you soon. Bye.